we're very excited to invite two speakers, and one is from HCA Africa, and then we'll also follow up from HCA Latin America. And really, this is the first time having them come and share with us their experiences of setting up their community in their region, and what are the exciting things that are happening, but also what are some of the challenges, and how we can learn and exchange notes so that we can really empower the local representation as well as research activities. So with that, I would like to introduce the first speaker of this session. So Colin Mr. Moranga okay. So he's a final year PhD student and a bioinformatician at the West African Center for Cell Biology of Infectious Pathogens, University of Ghana. And his PhD research focused on using flow cytometry and single-cell RNA sequencing to understand immunological and transcriptomic differences between clinical and subclinical malaria infections. So he's currently a member of the HCA Africa network in the planning committee. So, Collins, thanks so much for coming all the way to Thailand, and we look forward to your presentation. Let's please welcome him with them.
Thank you very much for the introduction. My name is Collins. I'm going to share with you about our experiences and lessons learned in setting up organizing the HCA Africa network. So efforts to develop the African component of the HCA in Africa stems back to the HCA Equity Working Group, which I think they started having discussions about setting up regional networks. And this led to a meeting in Addis Ababa in Ethiopia in 2019 with colleagues from African continent, from different countries. And this basically kickstarted the discussions about, having this Africa network. And the network has been expanding through the years, and there's hope that, African scientists can join and help in contributing to the development of the reference maps for understanding human health disease. So basically, the initial group that makes up the advisory committee is led by Gordon Awandari from Ghana, Nicola Mulder, South Africa, Musa Mwalanga, Alfred Ngwa in the Gambia, Christian Happy in Nigeria, and Prof. Thumbindungu. And we form part of the planning committee with the assistance from the HCA team. So basically, the initial gap was how do we reach out to scientists across the continent to start having discussions on how to strengthen the network. And with this, we conducted the first HCA Africa Symposium, which was held in March this year. And this attracted almost 136 unique viewers across 24 countries in Africa. And we held extensive presentations from individuals who are already conducting single-cell research in different countries in Africa. And basically, it also created an opportunity or a platform for discourse and awareness about the opportunities for joining different HCA biological networks, the Human Cell Atlas, equity, ethical issues, standards, and opportunities for collaboration with other HCA networks. And also, it's created an opportunity for starting to have, what is the future direction for single-cell research in Africa. And one of the ways that, that stood out was to hold workshops so that you can create this awareness to different scientists. And we got an opportunity to start having workshops, computational workshop, and this was conducted during the H3 Africa and presented to fellows who are already working in human health, heredity, and health, different diseases in the HCA, in the H3 Africa Consortium meeting. So we held a one-day workshop, And basically the learning objectives were to help them understand some of the steps involved in single-cell data analysis, learn how to visualize and explore single-cell data, and basically trying to understand how to use a differential gene expression to study different cell types. One of the key lessons we learned from this workshop was that, early career scientists, there was still a gap in understanding what is single-cell analysis about. So we went back to the drawing board. We decided we hold initial introductory workshops, and we started this. We made an invitation for application, and we had almost 200 people applying for an introductory workshop. And we decided to split it into two. We did an initial one of 90 individuals in October this year, and we intend to do others in November. Basically is to just introduce, single-cell RNA sequencing tools and techniques to researchers. And we are saying we don't really need prior experience because we are going to build up experience through the 4-day workshops. So we intend to hold others in November and the like. So in future, we hope that, we can start having in-person workshops whereby we also train experimental work as well as bioinformatics. And we hope to hold this. One of them that we're already planning is in Ghana in February 2023. And we hope to do others in Nigeria, in Kenya, South Africa, and Ethiopia. And we are really grateful to the Human Cell Atlas because they are actually sponsoring most of these workshops. And they are forming part of the initial training in experimental and bioinformatics. But into the future, we want to hold, refresher and advanced training workshops. And also, if possible, we can have mentorship and support program for scientists who are actually starting to use these techniques in their research. And we hope that, we can actually build on securing funding so that we can have postdocs who train on single-cell research and PhD or master students. And also, if possible, we can develop regional technology hubs for easy access and use of infrastructure that is required for single-cell analysis. So, apart from learning collaborations with Human Cell Atlas in Asia and Latin America, we also hope to learn how you all are securing funding for single-cell research in Asia, some of the strategies that you are using to get people involved, advances on experimental methods, how you're doing ethics and standards in single-cell research, public engagement, and equity and benefit sharing. So these are some of the things we are hoping that we are going to learn from all of you from the HCA network and regional networks. And I would like to thank the University of Ghana, where I am from, and West African Center for Cell Biology of Infectious Pathogens in Ghana, and the Human Cell Atlas Asia for inviting me here today, as well as the Human Cell Atlas for providing the funding to enable me to attend here. And as well as thank these people who have assisted in, conducting the workshops that we are already doing, as well as the future networks, workshops that we intend to do. Thank you very much.
Thank you, Collins. And it's very exciting that it's coming together very nicely in Africa, and really like a lot of these initiatives that are happening to support young investigators. Just maybe a quick question on this training workshop that you mentioned. Would love to hear your feedback on how it went. What were some things takeaways that we could all learn from? Because this is something that we also want to be considered for a little bit here, but it just got a little overwhelming organizing this meeting as well. So we would love to also in Asia have some workshops like that for computational analysis.
Yeah, I think one of the lessons that we've had is that, people are asking about, what are the infrastructure to conduct these single-cell experiments. So, and most labs in Africa, I think, because of probably lack of awareness of these technologies and what they can actually do, they haven't really equipped for most of these things. And that's one of the key things that scientists in Africa are asking, how can we do this? And probably this is an area that needs to be expanded on and build some of these infrastructures or provide collaborations with scientists in Asia to assist, as well as other regions. Yeah.
Great. So are you talking infrastructure as in like pipe data structure, infrastructure to servers, or are you talking more about?
Yes.
Yeah.
So
Okay.
both computational infrastructure
Correct.
to handle these data sets, as well as experimental part to generate these data sets.
Great.
Yeah.
Okay. All right. Well, thanks one more time. So I'm going to invite Partha here, who's actually co-chair of this session, and he'll introduce our next speaker. So, Partha.
So it's really my pleasure to introduce the next speaker, who has been a champion in HCA Latin America. Her name is Patricia Severino. She's a group leader at the Research and Education Institute in Albert Einstein Hospital in Brazil. She has a bachelor's degree in biological sciences and PhD degree in molecular biology. Her research group works on the integration of clinical and molecular markers, including microbiota and single-cell sequencing for the molecular characterization of cancer and treatment patterns of responses to various patterns of treatment, and on genome diversity and the comprehension of host pathogen interactions in healthcare-associated infections. Like I said, she's been a champion of HCA network, forming the HCA network in Latin America through the organization of local meetings and courses to engage researchers and other partners in the region. Patty isn't able to be here, but she's sent in our video recording. So Patty, go ahead.
Greetings, everyone. I would like to first thank the organizers for the opportunity of presenting our experience in Latin America in the Asian HCA meeting. So the title of my talk is Capacity Building Through Collaborative Networks for Single-Cell Analysis, the Latin American Experience. The Human Cell Atlas mission says that they aim to create comprehensive reference maps of cell types and properties of all human cells, and of course this demands a global effort. Back in 2019, the Equity and Diversity Working Group from the Human Cell Atlas understood that some regions of the world, including Latin America, were really underrepresented in this mission, and they wanted to understand why. So they put together a couple of meetings to discuss equality, so how these tools and ideas were being spread, right, for these different regions, and this was mostly inexistent in Latin America. And equity, so how could you bring together different regions, different communities, taking in consideration their local capabilities and regional interests to participate in the Human Cell Atlas initiative. Our first HCA meeting dedicated to Latin America was held in 2020, and at the time, what the Human Cell Atlas was, it was not really known in the region, right? So one of our main ideas was to introduce the Human Cell Atlas. So we presented the regional networks, including the Asian one, the HCA working groups. We looked for Latin Americans who were engaged in some kind of single-cell analysis project. We were able to find a few, but very few at a time. And one very important aspect at this time was the use of the breakout sessions. We went about creating breakout sessions based on the specific themes. So researchers, they could get together and discuss their projects and how single-cell analysis could contribute to what they were currently doing and how they could engage with the Human Cell Atlas. So themes, diverse themes, infectious diseases, cancer, specific questions from the region were brought about, and we carried out some massive announcements about the meeting, so we could really reach out for very different communities of researchers, right? Our most important take-home messages at the time were the lack of infrastructure for this kind of technologies, complete lack of funding for it, especially when we think about healthy human cells, and very minimal knowledge on the available technologies, which were available in many other different regions of the world. But we saw that there was a very high interest in contributing and participating. And the immediate action that we could identify at the time was training. So the following year, some of us got together to organize a first single-cell RNA-seq data analysis workshop in Latin America. Basically, people who met during the 2020 event. And the event was carried out in Mexico, Chile, and Brazil. The purpose of the meeting was to instruct researchers and students on how to design single-cell RNA-seq experiments, manage and analyze data. So during the first week, there was an overview of single-cell RNA-seq data analysis workflows with lectures from experts that were invited. The second week was a hands-on workshop using real single-cell RNA-seq data sets, not generated in Latin America because they were non-existent at the time. But the hands-on workshop was carried out simultaneously in Mexico, Chile, and Brazil. Each group in these countries was coordinated by local researchers in local languages, and the results were shared daily among the three groups. We believe the course was a success. We had some outcomes from it, so abstracts presented in meetings, including the HCA General Meeting of that year. Our second Latin American meeting was just happened this year, and the idea was to bring together this time more Latin American scientists exploring single-cell sequencing technologies for discussions of bottlenecks and applications. And a very important aim of this meeting was to give room for students and postdocs who wanted to discuss their work. So we had dedicated breakout sessions for them. We had very positive feedback from them. They were really happy, and they are eager to discuss the difficulties that they have and what they have already learned. Some of these students were students from the workshop carried out last year. So for us, this was a very big measure of success and interest in these kinds of events, right? And here I bring to you another success story, I would say, or a way to highlight how we have been working together in Latin America to be able to get funded, which is one of our most important bottlenecks. This is a project financed by Shan Zuckerberg Initiative within the Ancestry Networks for the Human Cell Atlas. Our project is called the Human Cell Map of Latin American Diversity. And what happened here, group of researchers from different countries in Latin America. Our coordinator is Andres Moreno here from Mexico. So Mexico, Colombia, Brazil, Peru, Chile, and in the US, researcher working with Latin populations. And so the team of the PIs are 100% from Latin America. And the way we went about was to look for groups that had expertises that could complement each other. So community engagement and sampling, the bench work, data analysis and bioinformatics, and training. We understand that Latin America represents a unique model for studying ancestry due to specific admixture between indigenous and other sources that contribute to the current population. And these ancestries are very much underrepresented in current research, right? Here is one example of data from one of the researchers in our group, and we want to explore this diversity at the single cell level. Additionally, we will address a specific problem from the region, which is gallbladder cancer associated with Mapuche ancestry in Chile. We will be working with blood, so PBMCs, also inspired by the Asian network, that since this is a kind of sample that all countries have access to, there are protocols that have been standardized also by, and with a lot of contribution by the Asian network. So besides the samples from gallbladder in Chile, we want to do then single-cell RNA profiling of immune cell types across these Latin American populations based on blood cells, create this new HCA bionetwork focused in gallbladder, and also create a Latin American community portal for single-cell information, data sharing, and visualization. The idea is to create this portal in Portuguese and Spanish to share not only single-cell data information, but also the aims of the project and the contributions that we believe will happen in a near future. The way that we are working, so all these communities and areas, regions are engaged. They have different interests and different local capabilities. We will process some samples in 3 different hubs, and we will be receiving students from all the different locations. A hub for sequencing in Brazil and one for genotyping in Mexico that will also be receiving students from the different locations. The analysis will be done jointly, and this can be done in a lot of ways virtually and converted into training and return of results to the community through visiting back the communities. What we hope to address with this project, which are the bottlenecks that we have identified through the meetings, and we are getting together to try to minimize them, which are bias in data sets, so improve representation of Latin American diversity within the HCA results, bias in leading teams, so improve diversity of researchers spearheading efforts, as opposed to just collecting and sending samples abroad for the analysis, improving or building local capacity for single-cell RNA sequencing. So through the hubs that we have created, we believe we will be able to gather students from the whole region to participate in protocols and analysis, and of course training in basic and advanced analysis pipelines, but also through our portal make the information available not only for researchers but also for the community in general. We believe that we came a long way from what was diagnosed in 2019 to our current community nowadays. Of course, it's not near what we see in the US or Europe, but we believe we are getting there somehow. We have been addressing regionally the issues of equality, meaning the distribution of tools and having contact with the ideas, and equity through bringing different groups together and talking and understanding what we can do and how we can leverage what we are not able to do yet, but how we would get there. So with that, I'd like to thank again the organizers, and I will be available for questions if you have any. Thank you.
So if anyone has any questions, I'm sure Patricia will be able to reply. Patty, thank you very much. That was a very clear exposition of what you're doing in Latin America, promoting HCA. There are no questions, but on behalf of all of us, let me thank you, and we hope that in our next meeting, you can be here in person. Thank you very much.
Thank you. Thank you, Paco.
Great. So we'll continue to our next 2 speakers who are been been contributor to the efforts that are being presented here for the past the day and the presentations forward. And for now, I'd like to invite Dr. Norbert Tavares. So Norbert, as we all know, who's here, is a program manager at the Chan Zuckerberg Initiative, CZI, who's part funder to this program. And just to give a quick introduction, previously Norbert was at AAAS Science and Technology Policy Fellow and Program Manager at National Cancer Institute at the National Institute of Health. In his former and current roles, he identifies and supports emerging and innovative solutions to biomedical research problems and develops and manages interdisciplinary grant programs. So specifically, he supports the development and dissemination of single-cell biology methods and its application to clarify the underlying mechanisms of disease. So with that, thank you for coming here, and we look forward to your presentation. Please.
Sawadee-ka. So thank you for inviting me to speak. Just a quick overview of CZI. Our mission is to support the science and technology that will make it possible to cure, manage, or prevent disease by the end of the century. So, pretty easy stuff. And the way we plan on doing that is by accelerating biomedical science, by developing new tools and technologies, and specifically around supporting open and collaborative models, right? And I think the COVID pandemic demonstrated how important open collaborative models are for moving us forward and advancing science. So we specifically have a program around single-cell biology. And why single-cell biology? Because we know that all diseases have a cellular mechanism for them. And so understanding them at the single-cell level is important for helping to get us to our goal. And we want to do this by accelerating single-cell biology methods, tools, technology, and resources. And the HCA is one of those resources that we see as fundamental to helping to move us forward, which is why we support it. So I was asked to talk specifically about our vision around equity and collaboration. And so I'll do that by telling you some of the things that we're doing now around these things, these areas. And so at CZI, we have been thinking about specifically racial equity and a set of questions that we want to use when building our tools and our programs in thinking about specifically racial equity in the context of the United States, but clearly that has relevance to the global community as well. And so those questions are, who is doing the science, what science is being done, how is the science being done, and who has access to this knowledge that's being generated. So our first data generating RFA forced us to think about the question of the who and who is contributing. And so on this map, the red dots represents where our grantees are located, and the size of the circle represents the number of grantees there. And it's pretty clear that we missed the boat a bit there. We're a new organization at the time, which is not a good excuse. And so we have done a lot of outreach to try to fill this gap and to not allow this to occur again. So that led directly into two of our more recent programs, the Ancestry Networks that you've heard a bit about and Patricia mentioned, and our pediatric networks. And so the goal of these was first and foremost to fill in these gap areas within the HCA around ancestry and pediatrics, but also to expand the representation of both the donors and the researchers that were involved in that. And the topic itself we knew would attract a number of groups, particularly international groups, which was important to us. And included in that is around both of these programs, requirements and encouragement to really get community input into the work itself and the questions that are being asked, and also return those that information to the communities that are donating the tissue. So this is what our current grantee networks look like. So we've improved quite a bit, but there are still some gap areas that we want to work on and support, especially in regions like Africa. So then there's the question of who contributes to the research. And so we tend to be focused very much on the who's in this room, but also the who's that matter are the tissue donors, right? And so we have had we're doing a few experiments now within CZI, one around a recent RFA that has closed, and it's funded now, but I can't tell you who the grantees are yet because we haven't announced it. And it's specifically around getting patient organizations to partner with research groups around rare pediatrics diseases. And then another experiment we're doing is with bridging the gap with underrepresented communities by having community-based organizations pairing with researchers, not on any specific project, but to help plan that project and get community input before there's a project, and then that project hopefully will go on to be funded. So we're doing all this work. We have these programs now that are generating all these data across a bunch of modalities and age and ancestry, the point being to build the Human Cell Atlas. And so those data are available now openly in the HCA data coordinating platform. Cell by Gene is a tool that I'll tell you about that we have those data available in as well. And then we've launched a program around getting insights from those data. So taking those atlases and learning new things and building new tools to help get more out of those data. So then there's a question of who has access to the data, what kind of questions can be asked, and who's going to benefit from these data. And so we've addressed that in a number of ways in our policies and the things that we fund. One really important thing is everything that is funded by us, all the data, code, protocols, have to be published openly. We encourage and we fund groups around protocols.io because if any of you have tried to replicate a protocol from a methods section, you know it's not quite that straightforward. We fund preprints because we want those data out open and early. And then the code as well. And then we've funded a bunch of tools around data sharing and discussion and annotation of papers and et cetera as well. So Cell by Gene is a tool that we have built within CZI. It's to enable researchers to be able to analyze the data without having to know how to code and to be able to make discoveries and filter the data. And there's a whole bunch of new features. If you knew Cell by Gene before, it's a lot different than it was a year or two ago. There's actually 2 posters. I'll mention it in a minute. And so it's one of the largest single-cell data sets, and it's not just healthy tissue, but also diseased tissue, because we collaborate with a number of other groups, and we want to get those data as a part of our system as well. So Cell by Gene is not made in a vacuum. We take a lot of feedback and interactions from you, the users, and we want your feedback. We want your questions. And if you want to figure out how to contribute your data, we want your data as well. So please get in contact with us. You can scan that QR code, email us. We've got a poster number 18 outside after this. Come and talk to us about it. Also, the data coordinating platform has a poster here, and those folks are here. If you could raise your hand. So find them if you want to know how to contribute your data. So it's great. You can have all of these data. However, if you don't have good quality metadata behind it, it's not very useful. And so one of the areas that there's a challenge with is around ancestry data. Half the data does not have any ancestry data associated with it or ethnicity. And so we've been funding a group to start to build out gaps within the ancestry. So for instance, here, this is the ontology for Africa. It's not looking great. And so one of the things we want to do is, as we're generating all of this rich data from our ancestry networks and pediatrics, is to have you, as you're generating those data, get in touch with the ontologist to start to really make a richer set of data. You don't know it yet, but you're going to get an email from me about this shortly, as well as existing ontologies to start to fill in these gaps and other consortia that are building these things so that we can have a better map that looks more like this. And then, the question of having all this rich data out there that nobody can use is not very helpful. So we fund the work that the Human Cell Atlas is doing around capacity building and doing these trainings. And I believe one of them, hopefully, will be coming to Thailand at some point, or Asia as well. And so what we really need, and is helpful, is to have folks on the ground, champions here. So if you're interested in this, find that guy there, John Randall, and we can help us build this out. And then, of course, you all, and meetings like this is critical, and building these collaborations, getting everybody in the room to talk about it, find out where those gaps are. And so funding these things and building these things and having these workshops is really important to us. So that's all I have. If you could leave this up during the questions. If you want to find out about our future funding opportunities, scan the QR code, visit that website, and here's one of them that's coming up in May and June.
Thank you, Norbert. Fantastic overview of CZI, and really appreciate your effort. There's some questions from the audience. Go ahead.
Yeah. Hi, Norbert. It's Ishan from India, Delhi. They have two questions. First question is there some thought given on the availability of the raw data, not just the processed data?
Yeah, so the DCP, the data coordination platform for the HCA, is where the raw data will live, and then the derived counts are in tools like Cell by Gene and other tools as well.
Okay. And is there any effort or thoughts around going after diseases that are endemic, such as what Patricia has mentioned about gallbladder cancer, things which are sort of endemic to areas across Asia, Africa, and Latin America?
Yeah, so the initial efforts right now are really around building that healthy reference atlas so you can have something to compare those diseases to. Eventually, we're going to start to move more into the disease space. Like, we've done some work recently around inflammation, because it's a really broad category of the mechanisms affect a number of types of diseases. As far as what the future holds as to what diseases, don't know yet. Part of that is building the local capacity to answer the questions that local groups want to answer, right? And so the Human Cell Atlas and doing the healthy reference is that first step. Getting everybody into the room is that first step. What Patricia showed about getting researchers together to start building the capacity to do single cell research for healthy tissue is that first step that we hope will lead to the next step, local funding to look at local diseases and interests.
So I'll be really interested in contributing in the gallbladder space, because that organ is currently missing in the...
Find me, send me an email, and I'll connect you to that group.
A quick question and a quick answer, please. Yeah. Over there.
Hi, I'm Elaine Dimokidis. I'm the CTO of Healthcare in APJ for Amazon Web Services. And at Amazon, we are thinking a lot about closing the gap for health equity, and we think that cloud computing is going to help towards that direction. And my question for you is, what are the efforts that you, from your side, you're doing to help fund the pioneers and the research labs that need to have cloud computing, and in order to collaborate and share data and actually do concurrent work together that it is going to produce insights? And do you feel that...
Please. Sorry. Quick question.
Yes.
So part of our funding model is that of flexible funding. So if you put it in the budget, we don't ask questions about it. We understand that computing costs are expensive and a part of the analysis work. And then we also fund, for our grantees, we fund a group called Lattice that the data they can put there, they can analyze it in a private space, and we help out a lot with the data wrangling and efforts around that. But we should talk.
Yeah,
Maybe
it'd be fantastic. Maybe Christine, quick question as well.
Very quick. Norbert, this might not be a surprising question to you, but it's great to build capacity on the ground. What about agency when it comes to management and governance of the data? And do you think that this kind of local agency is conflicting with fair data principles?
So that's hard, right? So that's the concept around data sovereignty is, I think, what you're getting at.
Exactly.
It's a new concept. We don't know how to implement
It's not new in the regions.
It's hard. We don't know how to implement it yet, and so we'll have to wait and see how it works out. I don't have a good answer for that because we just don't have good models in place yet to really see how to do that well.
Thank you.
All right. Once again, let's thank Norbert for a wonderful presentation.